Welcome back. In this second and final video for Chapter 22, we're looking at a few more structures found on the surface or inside prokaryotic cells, how they exchange genes with one another, and then again we're going to look at a brief overview of how they impact human lives. One of the structures we saw on a previous slide that some bacteria have is a capsule layer. And this capsule layer would be outside of the cell wall, and it's either a polypeptide or polysaccharide layer, or sometimes a combination of both polypeptides and polysaccharides that make up this jelly-like layer on the outside of the cell. These capsules help attach these bacteria to surfaces, for example, in our mucous membranes, to the back of the throat, or maybe to the reproductive tract. These help protect the bacteria from the host immune system and can even prevent these bacteria from being recognized by, for example, the human immune system. They allow them to be hidden, almost like an invisibility cloak for these bacteria. And in some cases, there are certain species of bacteria where there are strains. Some of them are pathogenic or disease-causing, whereas others are not pathogenic, not disease-causing, and the only difference between the two types is that one has a capsule, the pathogenic one, and the non-pathogenic one does not have a capsule. So here, these bacteria are more able or more likely to stick to our body surfaces and be harder to get rid of, harder for the immune system to get rid of. Whereas these, if they do not have a capsule, it may be easier to get rid of, and they don't have a chance to become established and divide in our bodies. Additional structures we saw previously include flagella. Singular is flagellum, but some bacteria have a tuft of these, a few of these at one end of the cell that allow them to move in some kind of aqueous environment. These are different from eukaryotes because they're much more simple in structure and they're made of a protein called flagellin rather than microtubules that we see in eukaryotes. And rather than the wave-like motion or undulation we see in eukaryotes, for prokaryotes they have a propeller-like spin that allows the flagella to move the cell around. Some bacteria also have pili, singular pilus, these little tube-like structures. These are also protein structures that are on the surface of the cell that can allow these cells to attach to surfaces and also allow conjugation to occur. And this is the exchange of genetic information from one bacterium or one prokaryotic cell to the next. Endospores are kind of a scary structure found in some bacteria. So some bacteria have the ability to produce endospores, which is when the cell is about to die because it's exposed to some kind of environmental stress. Let's say it's running out of space or running out of food, or there is some kind of harsh environmental signal, like extreme temperature, maybe change in salt concentration. When the cell is about to die, what it does is it produces a thick protein wall around its DNA, and the rest of the cell will die, will break down, releasing the endospore, this green structure here, into the environment. Since it's only really protein, a protein shell around the DNA or the genome, it doesn't need any nutrients, it doesn't really need anything to survive. It can be very resistant to environmental stress and even heat. In fact, if I have something and I think it's contaminated with endospores, even if I boil it for 30 minutes, these endospores can still survive. They can often survive even bleach or even our harshest cleansers. When the endospores go back into a favorable environment, perhaps through humans accidentally ingesting the endospores, they're in a nice warm environment, there's plenty of food for them. What happens then is they germinate. That means they become vegetative cells again, they metabolize, they start dividing once again. The problem is when they germinate, these endospores turning back into normal cells, they release a lot of toxins, and these toxins can be deadly. So the types of bacteria that can produce endospores include the bacteria that cause these problems like tetanus, botulism, and anthrax. Continuing on, remember our nucleoid is the region where we're going to find our main chromosome. In this area, sometimes we also find extra chromosomal DNA, those small circular plasmids. We have also ribosomes in the cytoplasm. These are different from eukaryotes because they are composed of different RNA and protein complexes. Antibiotics are drugs that kill bacteria, and some of them work by attacking prokaryotic ribosomes. And this is nice because since eukaryotic ribosomes and prokaryotic ribosomes differ in their structure, antibiotics can specifically attack prokaryotic ribosomes without harming our eukaryotic ribosomes. Remember, antibiotics are not effective against viruses because these are usually, almost always, created or naturally formed to attack specific structures on bacteria rather than structures on viruses, which are completely different. Looking at prokaryotic genetics, how do bacteria divide? So all prokaryotes divide by binary fission. What is that? So what happens first? Let's say this is a bacterium, and we have our original parent cell. The first thing that happens is it makes a copy of its chromosome, its DNA, and at the same time, the cell is getting a little bit bigger. The cell continues to grow and elongate and forms an invagination between the two. Eventually, the cell wall formation is complete, and you have two identical daughter cells. Remember, this is not sexual reproduction since the daughter cells are genetically identical. So how do bacteria, for example, increase their genetic variation over time to survive varying and harsh, sometimes, environments? They can go through horizontal gene transfer, exchanging DNA with cells from the same generation. And they do this through conjugation, transduction, and transformation. We'll go over each of these in detail. This is seen in bacteria, but also seen in our archaea. This slide shows us the three types of horizontal gene transfer in prokaryotes. So the first one on the left is transformation. In transformation, bacteria are taking up DNA directly from the environment. So here's my bacterium. It could be naturally found. This DNA could be naturally found in the environment, or it could be artificial, where I have the bacterium in a test tube and I add some plasmid to the test tube. So here I have some plasmid DNA, either from another cell or from the environment, and the cell takes up this plasmid and incorporates it into its own genome, into its own chromosome. The second one is transduction. Transduction requires bacteriophage. So a phage is a virus, a type of virus, and bacteriophage are viruses that specifically infect bacteria. So bacteriophage do not, for example, infect human cells. Here I have a bacterium. This is the bacterial cell with its chromosome. And here is the bacteriophage. The bacteriophage will bind to specific receptors found on the bacterial cell, bind to it, and inject its viral DNA into the bacterium. This DNA could have been picked up from a previous infection of another bacterium. So if I had a previous bacterium over here and it had its own chromosome, what could have happened is this virus could have infected this bacterium, picked up some of its genetic information, and it could be injecting some of that information into this new cell. And I can see that this bacterium has incorporated the DNA from the bacteriophage. In the last one, C, conjugation, I have two bacteria. One of them on the left has a plasmid, and I can see the one on the right did not have a plasmid. So it looks like the one on the left is forming this tube-like structure, which is the pilus, allowing a copy of its plasmid to be sent through to that second cell. And at the end of this process of conjugation, both of these bacteria will have a copy of that plasmid DNA. So plasmids usually encode something advantageous to the bacterium because it's extra chromosomal DNA. This DNA is not needed or not necessary for normal survival and function. In E. coli, for example, the bacteria shown in this picture, conjugation really depends on whether the bacterium has something known as the F plasmid, which contains the fertility factor. The fertility factor allows the cell to form a pilus, the tube, connecting it itself to another cell. So F plus cells contain fertility plasmids and F minus cells do not. And these can only exchange information with each other. So an F plus cell cannot exchange plasmids with another F plus. It has to go from F plus to F minus. So the fertility factor will allow the cell to create the pilus, this tube-like structure. The cell will make a copy of its plasmid and send that copy through to the F minus cell. And at the end of conjugation, it's pretty interesting because now the cell also has a fertility plasmid, or the F plasmid, and now it becomes an F plus cell. Here's another look at that same process in a little bit more detail. We have our F plus cells on the left, F minus cell on the right. The F plus cell is the one with the fertility plasmid that can form that F pilus. The F plus cell will make a copy of its plasmid using an enzyme we'll look at later on in the course called DNA polymerase, and it'll send that copy of the plasmid over to the F minus cell. If the entire plasmid is successfully transferred in time, then in time meaning while the cells are still connected to each other, because sometimes the cells break apart from each other before the transfer is complete. If that happens, the cell stays as an F minus cell. But if the transfer does finish in time and the recipient cell receives the entire F plasmid, now this is an F plus cell at the end. Why is this important? This is important because there could be certain beneficial characteristics found in that F plasmid that this recipient cell previously did not have, but after the exchange, this also now has those advantageous characteristics. One of the other processes we talked about of horizontal gene transfer was transformation. Sometimes this is natural, and this is when a bacterium, let's say this is my bacterium, my normal chromosome, will sense that there's extra chromosomal DNA in the environment, and it'll pick it up. And it can stay as a plasmid form, or sometimes it becomes integrated into its own chromosome. And this is neat because it allows the bacterium to, again, pick up additional or new characteristics it previously did not have. We also have artificial transformation, which happens in the lab. And we're going to be doing something like this towards the end of the semester in our lab. So let's say I have some kind of sensitive bacterium over here, antibiotic sensitive bacteria. Let's pretend this bacterium is sensitive to the antibiotic penicillin. What that means is penicillin can easily kill these bacteria normally. What I'm going to do now is try to turn these sensitive bacteria to resistant bacteria. Bacteria that can grow even in the presence of penicillin. So I'm trying to generate penicillin resistance. What I can do is take my sensitive bacteria, put it into a test tube with plasmids that contain the penicillin resistance gene. These sensitive cells can be forced to take up DNA, usually through some kind of electrical shock or adding ions such as calcium to disrupt the cell wall and cell membrane to make it porous. So these cells temporarily open up and take in that plasmid. Now I can see my sensitive cells have taken up that penicillin resistance gene from those plasmids. And now these cells resist penicillin and can still grow in the presence of penicillin. So we will be doing something similar to this in lab, and we often do this with E. coli when we're trying to clone certain genes. We clone genes in bacteria because they divide very quickly and they easily make copies of the DNA of interest. So that plasmid I just talked about is a type of resistance plasmid. Resistance plasmids are plasmids that carry antibiotic resistance genes, allowing the bacterium to become resistant to a specific type of antibiotic. And we see this a lot in different types of bacteria, such as Staphylococcus aureus. You may have heard of MRSA, methicillin-resistant Staphylococcus aureus, or even MRSA. Vancomycin-resistant Staphylococcus aureus. These are bacteria that have over time acquired many different types of resistance plasmids, making them resistant to a broad spectrum of antibiotics, unfortunately. We also have plasmids called virulence plasmids, which are plasmids that contain genes that code for virulence factors. These are factors that allow bacteria to invade and spread and cause infections in the body. Transduction through those bacteriophages, remember those viruses that infect bacteria, these phages can also include genes that cause harm or allow bacteria to become pathogenic, disease-causing. One example is in a family of bacteria found in our GI tract, in our intestines, called Enterobacteriaceae, which include E. coli. Most E. coli bacteria are harmless. We have our E. coli, its normal chromosome. It doesn't really cause problems in the body as long as you wash your hands after going to the restroom and before handling food, for example. But E. coli strain 0157H7, what happened is one of these bacteriophage infected E. coli and injected DNA encoding virulence factors that produce toxins that are deadly to humans or can be deadly to humans. So this strain now produces a toxin that can cause very strong symptoms and sometimes death in people who are infected. And it wasn't that long ago, even just in the early 20th century, that infectious diseases used to kill a fifth of children before they turned 5. So sanitation, proper hand washing, proper aseptic technique, avoiding contamination, and the use of antibiotics really improved the situation. But more recently, because of the abuse of antibiotics and the overuse of antibacterials, bacteria have become resistant to antibiotics, really through the spread of those R plasmids, through transduction, and the spread of resistance genes, which is especially prevalent in convalescent homes and in hospitals. If we look at some examples of how specific microbes impact our lives, one common example is dental caries, or cavities. So our plaque buildup throughout the day includes bacteria in their biofilms, so a bunch of bacteria clumped together that secrete this sticky layer known as a biofilm, which attracts even more bacteria to the area and allows them to stick really well to our teeth. These biofilms include the bacterium Streptococcus, including other species as well, but one of them is Streptococcus, which ferments any sugar in the area to form lactic acid, and that lactic acid can cause the degeneration of our tooth enamel. And we didn't know this before, but sharing food, sharing things like toothbrushes can actually spread bacteria from one person's mouth to someone else. So nowadays, usually adults are discouraged from sharing food with young children who don't have bacteria established in their mouth yet. Their oral cavity, their oral bacteria is not established. So if a parent has a likelihood or a tendency to get cavities and they share food with their young children, those bacteria can be transferred to the children, causing them to be also more likely to get cavities. Another bacterium shown here is Helicobacter pylori. This is the causative agent of ulcers. And it's really interesting if you have time to read about it. The two scientists who won the Nobel Prize for this discovery, don't try this at home, but one of them actually drank this bacterium, a petri dish full of Helicobacter pylori, to prove that it caused ulcers. And these are treated with antibiotics because this is a bacterium. One of the ways we can slow down the spread of bacterial diseases is through vaccinations. So vaccinations usually involve the injection of an inactivated form of the bacterium or virus. In this case, if we're talking about bacteria, they usually don't give you live bacteria. It's usually a piece of the bacterium, such as a protein from its membrane, that you can get injected into your body, and that acts as a flag, so that if your body encounters it in the future, it knows to attack this foreign substance. Communicable diseases, these are diseases that can be spread from person to person. Contagious diseases are very communicable. They're very rapidly and easily spread between individuals. And as I mentioned at the beginning of this chapter, even though we tend to focus on the bacteria that cause diseases in humans because of the problems that arise, there are actually many more beneficial prokaryotes out there, including the ones we use to make foods for us, including cheese, wine, beer, yogurt here. And we're going to look at fermentation and the production of alcohol, for example, in our second exam material. Finally, when we look at the types of relationships we have with prokaryotes, there are three types. We have mutualism, where both parties benefit, us and the bacteria, for example. Commensalism, where one member benefits and the other one is not benefiting or harmed. And then parasitism, which is when the parasite benefits and the host is harmed. So we have these all over our bodies in terms of prokaryotes. Mutualism includes, for example, the bacteria on our skin. These are good bacteria. They prevent harmful bacteria from taking over by competing with harmful bacteria for space and nutrients. Commensalism, a lot of the times the bacteria in our GI tract, in our intestines, are called commensals because theoretically they don't harm us and we don't benefit from them while they get a safe space to grow and lots of food. But really it's more like a mutualistic relationship because some of our bacteria in the intestines actually produce vitamins for us. And then parasitism could be something like the bacterium that causes tuberculosis. The bacterium benefits, the parasite benefits, but we are harmed because of the disease. And that takes us to the end of chapter 22. In our next set of videos, we'll cover chapter 5, which looks at the structure and function of plasma membranes. All right, thanks for listening.